So we're going to be talking about a cancer drug, TR100, which basically targets an integral or an important part of the cell structure, the microfilaments in cancer cells. The drug is currently in phase 1, so there are no clinical trials or human clinical trials that have been released to the public. However, this drug has been treated or tested in vivo in mice and in vitro in mice and human cells. So, could we have a volunteer? Jack, back it up. So, this is a person with a fully functioning spine, right? I think we need another volunteer. Sarah, will you come up? No. Sarah, right over here. Could you please demonstrate to us what a person without a spine would look like? So clearly you can see that without a structure, without a structure, which in the analogy was a spine, the cell can't function or survive. So stand up. Let's give a round.
Wait. I want to make sure that I understand mechanism, and we're going to definitely pause the time here just to make sure that we take the time to truly appreciate the point that they're trying to make. I'm sorry, could I see the difference between the spine and the no spine one more time, please?
Sure, okay. So Jack represents a person with a fully functioning spine. Sarah, could you demonstrate to us what a person without a fully functioning
guys, do you have any questions? Frankie? I'm sorry, can you raise your hand? Go ahead.
I think I have my eyes closed, so.
Yeah. I think
maybe a slow motion. We need to see it again.
I think a slow motion would be appropriate. Karthik, if you wouldn't mind.
slow motion?
Yeah.
Am I supposed to go in slow motion? No.
But you want to say her do it in slow motion.
Alright. Jack represents a person with a fully functioning spine. Sarah, can you demonstrate to us what a person
In slow
without,
motion.
in slow motion, what a person without a spine would look like?
Alright, okay, time's back on.
Alright, let's give a round of applause. Let's give a
Okay, so we know now that a cell without a cytoskeleton will collapse and fall just like Sarah did. Well, there's three main systems within the cytoskeleton that we must learn before we can understand the mechanism behind our drug. So, microtubules, intermediate filaments, and microfilaments. Now, microfilaments is the main one that we will be focusing on, and microfilaments are made up of two proteins, actin and tropomyosin. Everybody got it? So we know that actin is a protein that makes up cytoskeleton, but what is tropomyosin? So tropomyosin binds with actin subunits to create a stronger microfilament. So if you had a microfilament made of just actin, it would be much weaker than actin and tropomyosin together. Now, there's many different isoforms of tropomyosin. There's been TM1, TM2, TM3, but the main one we're focusing on is TM5 and M1.
What is an isoform?
Isoform is it's a protein that is expressed differently, but it has the same genetic information. So what we will be targeting is the TM5 and M1 gene. And as you can see here, the orange strand is tropomyosin, and these are the actin subunits.
Oh, wait, yes.
Wait, so you're saying that isoform has the same genetic information but it's expressed differently?
Yes.
So would epigenetics play a role in that?
Isoforms are similar to
So just think of it like
yeah.
this. Right there, like, so they all have like very similar components, but they have like small functional groups that are modified in each one. So they're very similar molecules, just have small.
What's they?
Oh,
protein,
the ice,
the
yes.
isoform.
Isoforms in general, all of the specific molecules. This base molecule is tropomyosin.
Do they have the same structure?
No,
They have
they're
slightly different structures,
Slightly
which is
different.
what helps the TR100 target drugs.
So they're all forms of tropomyosin. They're all tropomyosin proteins, but they have different names because they differ very slightly.
Okay.
You said they're just slightly different in shape. Is there a functional difference as well?
Yes, since shape determines function. If it's a change in the shape, then yes, it'll
I'm sorry.
have
Did they clearly define what the change in shape, the change in
Yeah.
function was?
Yeah.
That's all I wanted.
next slide. So,
This is, so now we're going to be talking about, right, we talked about different parts of the structure. So why are we targeting tropomyosin instead of, let's just say, microtubule? Well, before this new class of drugs, anti-microtubule drugs were trying to target the microtubules in cancer cells. However, the drugs weren't able to successfully differentiate cancer cells from healthy cells, which made it less effective. However, because there are different isoforms of tropomyosin and certain tumors, that this TR100 has been tested on, had certain isoforms which are found in higher levels in cancer, we could target those. We could target the TM5NM1, which is a form of tropomyosin found in largely in tumors, to successfully really break down the structure of the cancer cells.
okay, so we know that tropomyosin has many different isoforms, and we know that there's the main one that we're focusing on is TM5NM1. So why do cancer cells have more? Well, as cancer cells simplify and adapt their structures, they become heavily dependent on tropomyosin, particularly this isoform. So it acts as a building block or main structure of the cytoskeleton. So how do they do this? Well, cancer cells, let's have a refresher first. siRNA is the process within a cell where the protein dicer destroys any fishy copies of RNA. So here what we're looking at in this graph is this is control siRNA in a non-cancer cell. This is TMN1 to siRNA within a cancer cell. So what we're looking at here is the amount of, or level of siRNA. So as you can see, there's much higher amount of siRNA within regular cells and a much lower amount of siRNA for TM5NM1 within cancer cells. So what this means is that if cancer cells are able to reduce the amount of siRNA for TM5NM1, then they're allowed to express or overly express the amount of TM5NM1 within the cell. So by reducing the amount of siRNA, they are able to overexpress this gene. So the reason why we're showing this is for you guys to understand that cancer uses the cell siRNA to reduce that to create more levels of TM5NM1. Next.
What specifically is the siRNA?
The siRNA is the mechanism which dicer proteins destroy any fishy copies. So fishy copies can entail overexpressed genes or incorrectly expressed. So by cancer cells reducing this process, they're able to overexpress this gene.
How do they target this like that siRNA?
We are not 100% sure how cancer cells reduce the amount of siRNA, but what's important to get from this is that the levels of siRNA within the cancer cells for TM5NM1 are reduced. That way they can create more tropomyosin. So that's the main point to get away from this slide, is that we want cancer cells need more tropomyosin than regular cells.
Okay, so using that information, we can look at these graphs and see how TM5NM1 is related to cancer and the effect of TR100 on this data. So in this diagram is plot maps, and these are on the top are certain names of different cancer cells that were tested for levels of certain proteins and building blocks and microfilaments. Okay, so So found in all of these types of cancer cells was TM5 and M12. Also listed were other isoforms of tropomyosin like TM16, TM2, TM3, TM4, and actin proteins that we have. So this proves that TM5 and M1, this specific kind of tropomyosin, is found in these listed cancer cells here. Here are mostly neuroblastoma and melanoma cells. Okay, so knowing that, we can move on to this graph right here. This shows the levels comparing this isoform of tropomyosin to this one, TM5 and M12, which is the one that TR100 is targeted towards, and TM2, which doesn't target towards. So, as you can see, the black bars represent the control groups of cancer cells that are not treated with the TR100 drug. And the gray bars show the levels of whatever tropomyosin after the treatment of TR100. So as you can see, for TM2, there's basically no difference before and after treatment of TR100 drug. But for TM5 and M12, it's greatly reduced, and you can see that there. This proves that TR100 has an effect on TM5 and M12 and decreases its level of cancer cells. Yeah.
Were they expecting a drop in TM2 levels after treating you with TR100?
No, they were not, because the main target is TM5 and M1 because cancer cells overexpress this gene. So by this graph, it's basically showing that TR100 specifically targets this isoform over this one. So it shows how it's pretty, TR100 is very effective in how it targets the tropomyosin. Yes.
How does it target TM5N and 1, 2,
Oh,
3?
yeah,
We're gonna get to that
Okay.
next.
Okay, so very good question. So what is the main mechanism behind this? Well, in this photo you can see the green part is the protein tropomyosin, TM5 and M1, and the purple molecule is TR100. So what happens is TR100 binds to the binding sites of tropomyosin, therefore tropomyosin can no longer bind with other tropomyosins. So what does this mean? Well, if you inhibit the binding site of tropomyosin, then it can no longer strengthen the cytoskeleton, or more specifically the microfilaments. Therefore, the cytoskeleton becomes weak and it will collapse. So TR100 inhibits the binding sites of tropomyosin, and therefore can no longer bind and strengthen the cytoskeleton. So this is the pathway. TR100 inhibits TM5 and M1, which weakens the microfilaments, therefore the cell structure collapses, yes.
You know how long it lasts for?
That was not specified.
It was not specified. And so now Claire will just go to another mechanism.
Okay, so just to clarify, right? This is showing how TR100 specifically targets the structure of the cell. Now, scientists have also found data showing that TR100, okay, causes an increase in PARP, which is basically a type of enzyme which activates caspase 3, which is also an enzyme which initiates or induces apoptosis or stops the cell cycle. But we don't have proof of how TR100 does this in cancer cells, but not in healthy cells. But we do have data, as we'll see later in the presentation, showing that the size of the cancer reduced, the tumor, which TR100 is targeting.
Okay, so this is a graph showing the tumor size after TR100 treatment. So this is zooming out a little. This is actually measuring the volume of tumors that's in the mouse, in the human neuroblastoma cells. So as you can see, the dark line is the control group. It's the group that has not been treated, and the light dotted path is the graph showing time after being injected with TR100. So as you can see, there is a drop in the amount of tumor found in these neuroblastoma TR100, and then tumors in that particular
Okay. This answers the question. Josh,
Do you know what the control group is? Or is it?
just normal.
It's just
Normal cells.
It's just normal
human
tumor
cells.
group. It's not treated with anything. It's just tracking the normal growth.
Okay. Which were tested in vitro, not in the laboratory. Yeah, good.
So TR100 was injected, or time post-cell injection. Was that injection of TR100? Or
Yes.
What is in vitro again?
In vitro it means behind the glass, so these are in cell cultures. This is not in an animal. This is just
In the lab.
I'm a little bit confused as to how the tropomyosin is related to cancer.
Oh, okay. So tropomyosin is what we're targeting, and so what's your specific question? How tropomyosin relates to cancer?
Yeah, like how the cancer is caused by it.
Okay, so the cancer cell, when it simplifies and adjusts its structure and to survive, in order to survive, it has to create more tropomyosin. So what this can mean is that the cell will strengthen its structure. So cancer cells need more tropomyosin and a stronger structure than regular cells. We do not know necessarily why, but we know that we have seen results that, like in the earlier graphs, you can see levels of TM5 and M1 were higher than in regular cells, and that's just because cancer cells need, that's just what they do.
Yes.
Is it possible that cancer cells grow resistance to the TR100 drug?
Well, since it's only in phase 1, it's not really known.
Do you know what happens after the 18-day period?
Sorry.
Do you know what happens after the 18 period line? The 18-day period line?
No.
there was nothing showing that.
Yes.
How would this affect normal cells? Like, I mean, you said that cancer cells need more tropomyosin, but like, would it still normal cells?
So TN5 and M1 too, right? Because cancer cells have lower, they're the ones that have been tested in the study, have lower levels of siRNA against TN5 and M1 and 2. The cell, because the siRNA kind of helps to regulate that expression, there's an overexpression on TM5 and M1 and 2. So now there's a higher level of those types or isoforms of tropomyosin that are in the actin filaments versus in healthy cells, there's a very small or a smaller number of tropomyosins.
So at certain dose
I think what Adam is sort of asking is why, even if there is a smaller amount of tropomyosin in normal cells, why is TR100 targeting the higher?
Yeah, why is
Could there be side effects?
Yes, there could be side effects, but since the dosage was quite small, it was only enough to affect cancer cells and high expression in cancer cells over the blood. So since there's such high expression within cancer cells, and I think this is what Carter was getting to,
Careful.
is that the dosage that they used, or they specified they tried many different times with different dosages and experimented, and this one didn't have as much effect as the other.
Did they look to see what would happen if after treating a tumor with TR100 for a given period of time, if like how long it was like after going off of the treatment, it would start to grow again? Or did they do anything?
Wait, so Cam just talked about they did the same thing with different dosages. What were the responses with different dosages?
So we have more graphs at the end of it, which are experiments with other types of cells, and you see different like slight variations. But these were the only graphs that were resulted because they took this is just the graph that showed this exact dosage. We don't necessarily know what other.
How is TR100 administered?
Okay, so in mice, when they tested in mice in vivo, it was administered through IV or injection.
Okay.
Let's keep moving on. We can ask questions at the end.
Alright, mouse melanoma cells and keeping in mouse neuroblastoma cells. So what was shown in the graphs was generally the result. And these, they were also tested in livers and hearts of mice in vivo. So like, a lot of the mice were living, this would be tested there. As you can see in this diagram, this compares the neuroblastoma cells before and after being treated anti-trophic myosin cell. So as you can see, the cell has just like shrunk like a lot. And the cytoskeleton, this like filaments here, you can see it having like deteriorated.
So, why do we care about this work? Well, it's it's a step forward because, like I said before, right, we're moving from anti-microtubule drugs, which are trying to target microtubules in the cell, break down the structure in that way. But we found out, those drugs weren't able to successfully differentiate between, it was killing both human and cancer cells. What we found, this study has found
Okay, thank you. That's time.